In this lecture, I will compare all the three complement pathway: classical complement pathway, lectin pathway, and alternative complement pathway. So the first is classical pathway, the next is lectin pathway, and the last is alternative pathway. In case of classical pathway, the first thing is antibody-antigen complex. So antibody and antigen complex start the classical pathway. In case of lectin, this is PAMP, PAMP. PAMP means pathogen-associated molecular pattern. This is actually the signature molecule of microbes. As the name of the pathway suggests, lectin means carbohydrate. So the PAMP like mannose, sugar, fucose, sugar, and acetylated oligosaccharide can start the lectin pathway. Lectin means carbohydrate. This pathogen-associated molecular pattern are the signature molecule of microbes. This mannose, fucose, and acetylated oligosaccharide can be present in the cell wall of bacteria and also some fungi. And in the alternative pathway, there is no actually any initiator, but the C3 protein actually starts the process. I will discuss about this. Okay, the C3 protein actually present in our serum in high concentration, and the C3 protein hydrolyzes spontaneously and it becomes C3H2O. So it becomes hydrolyzed very rapidly. This process is called takeover. So it becomes rapidly or spontaneously hydrolyzed and forms C3H2O. So in case of alternative pathway, this is the initiation. As you can see, as this classical pathway requires antibody, so this is actually in the adaptive immunity. It falls in the adaptive immunity. In the lectin pathway, this PAMP is actually falls in the innate immunity. Our innate immune system expresses some receptors that can recognize PAMP, like toll-like receptor, Rig-like receptor. So this PAMP is actually under the innate immunity. So the lectin pathway falls in the innate immunity. So now you can see this complement pathway actually cooperate between adaptive and innate immunity. Okay. And in case of alternative pathway, as there is no actually any initiator, so we cannot call it any adaptive or innate immune system. So there is no explanation of this. Then the third point is initiator protein. This is the initiation process. When antibody and antigen recognize itself, then the process starts classical pathway. When PAMP means any microbes enter our body immune cells have some receptors that can recognize this PAMP. So this is the initial mechanism, and in the complement pathway there are initiator protein that initiate the pathway. Like in case of classical complement pathway, the initiator protein is C1q protein. This is the initiator protein. This C1q can recognize the antigen-antibody complex. Specifically, I want to mention C1q recognize the CH2 domain of antibody. Next is in case of, so this is the initiator protein, and in case of lectin pathway, the initiator protein is MBL, mannose binding lectin, and in very few recent years it has discovered another protein called ficolin. This MBL and the ficolin and also the C1q protein acts as a opsonin. This MBL protein, mannose binding lectin, can bind with high affinity mannose and with high affinity fucose, and the ficolin actually bind with acetylated oligosaccharide. Next is in case of alternative pathway, you can say the initiator protein is actually C3 protein. Now the fourth point is there is an enzyme that actually responsible for the cleavage of complement proteins. In case of classical complement pathway, the protein is called C1R and C1S. These are the serine protease. C1q protein is actually associated with these two partners. They are present 2 number, means double. This enzyme actually cleaves the complement proteins. In case of lectin pathway, the enzyme is called MASP123. MASP means MBL associated serine protease. The same function performed by the C1R and C1S is the MASP123, and in this case I want to mention the number 2, MASP2 is very similar in structure with the C1S enzyme. This MASP123 actually shared by these two protein, MBL and ficolin. MBL also expressed or attached with this protein, so it is the partner of MBL, and ficolin also contain MASP. In case of alternative pathway, the enzyme is that actually cleaves the complement pathway initially, that is the factor D. The very important point is another thing is C3 convertase enzyme. What is the C3 convertase enzyme in this classical pathway, lectin pathway and alternative pathway? In case of classical complement pathway, the C3 convertase enzyme is actually C4b and C2a complex molecule. So this is the C3 convertase in case of classical pathway. This C3 convertase enzyme cleaves C3 protein into two fragments, C3a, C3b. So this is the C3 convertase enzyme. In case of lectin pathway, the C3 convertase is same as the classical complement pathway. This is C4b and C2a complex. But in case of alternative pathway, there are two C3 convertase. One is called fluid phase C3 convertase, that is C3H2O, means hydrolyzed C3 with BB protein. This is actually factor B. When it cleaves by this factor D, it becomes two fragments, B small b, capital B small a. This capital B small b actually the larger fragment that attach with this C3H2O, means hydrolyzed C3, to form the fluid phase C3 convertase. But in case of alternative pathway, there is another C3 convertase that is very stable. This fluid phase C3 convertase is very unstable. It actually synthesizes rapidly and degrades also rapidly. But if there is any infection, then another type of C3 convertase is made, that is called stabilized C3 convertase. This stabilization is performed by another factor called properdin. The stabilized C3 convertase is different. It is C3b and BB complex. Another thing is 6, that is called C5 convertase. This enzyme in case of classical pathway is C4b, C2a and C3b. So this is the C5 convertase. In case of lectin pathway also, this C4b, C2a and C3b is the C5 convertase. This enzyme can convert or fragment the C5 protein, complement protein. When the C5 gets degraded or cleaved, it becomes two part or two fragment called C5a and C5b. So this classical pathway and the lectin pathway merge into the C3 convertase enzyme and C5 convertase enzyme. And in case of this alternative pathway, the C5 convertase is C3b, BB and also another time C3b. But after this formation of C5 convertase, all the pathway actually merge. All this pathway actually cleave the C5 protein into two fragments, C5a and C5b. This is the smaller part and this is the larger part or fragment. The smaller fragment acts as a anaphylatoxin that actually induce inflammation, attached with another complement protein called C6. When the C5b and C6 combined, then it actually recruit into the membrane or the wall of the bacteria. Then what happens, this C7 actually attach with C6 attached with the C7, then C7 attached with C8, This is a chain reaction, and after a protein called C9 gets attached, and multiple of C9 form a complex that is the ultimate product of these three classical pathway, lectin pathway and alternative pathway, that is called MAC, means membrane attack complex, that form a pore-like structure in the bacteria, and eventually the bacteria die. So this is all about the comparison between classical, lectin and alternative pathway.